Well, I would like to welcome everybody who decided to join us for Sano seminar on computational medicine. And today we host as a speaker, we host Philipp Harth. Philipp has got a Master of Science degree in computer science from Heidelberg University. And then he was software developer at Carl Zeiss and Charité. It was linked with Medical University in Berlin. And currently, Philipp is a research associate at the Visual and Data-Centric Computing Department at SUSE Institute in Berlin. And well, his current interests are visual analysis tools, and he's working on computational network models to predict connectivity, anti-difference assumptions of different type of, let's say, illness in brain. Well, if you will have questions during Philipp's talk, please put them on chat or on questions and answer. I will try to read them. And well, and then, as usual, there will be a discussion after the Philipp talk. So, Philipp, please, screen is yours.
All right. Thank you for the invitation and the opportunity to share my work here. I will talk about brain network models at the microscale, visualization and modeling. Let me briefly give an outline of my talk. First, I would like to give a background about computational connectomics, what are the goals in this field, and what is the project that we are working in. Then I'd like to focus on a computational model we built of the red barrel cortex, and it's called barrel cortex in silico. And then I'll present some ongoing work I'm currently involved in about generative network modeling to predict synaptic connectivity. And one of the areas of research in our department is interactive visualization. So this will be the last part of my talk, and I'll finish with a brief conclusion. Let me give you some background about the project that I'm working in. It's a cooperation project between 3 labs, the Max Planck Institute for Neurobiology of Behavior in Bonn, and specifically the in silico brain sciences group of Professor Marcel Oberländer. They're doing experimental neuroscience, but also computational neuroscience, trying to understand the neural basis of behavior. We're also cooperating with a machine learning group in Tübingen, headed by Professor Jakob Macke, who is developing inference tools, machine learning-based inference tools for the natural sciences. And I myself, I'm affiliated with Dr. Daniel Baum at the Visual Data Analysis Group at SUSE Institute. And as said, our focus is visual data analysis, and our role in this cooperation project is to facilitate our domain experts, that is the neuroscientists at the Oberländer lab, in understanding the data better and providing the computational means to conduct their research. This cooperation project is part of a larger consortium, the SPP 2041 Computational Connectomics program, funded by DFG. And we're now in the second funding period and about to conclude this research program. And most of the work I present here is, in fact, a cooperative work with these 2 other labs. To give some background about connectomic research. The goal in connectomic research is, briefly said, to understand the wiring diagram of the brain. And the brain is a multiscale network. It's organized at different levels. At the macro scale, you have long-range connections between different brain regions. These are myelinated axon bundles that realize long-range connections between different areas of the brain. At the mesoscale, you could say you have individual cells, that is neurons, which are shown here on the left, with their cell body and the complex branching structures that emanate from the cell body. And these neuronal cells are highly compartmentalized, consisting of the cell body, also called the soma, the dendrite, which is collecting incoming signals, and the axon, which is conveying signals that are forwarded to other neurons. It's more complicated in other, this is for cortical neural networks, for example, in the insect brain. This compartmentalization into dendrite and axon is usually not as clear. And at the finest or highest resolution level, you have synaptic contacts at the nanoscale, which are the connection points between neurons conveying signals from one neuron to another. And typically they connect one axon to one dendrite. Now, we are mostly, as the title indicated, working at the microscale, so we are interested in these neuronal cells. We are not doing this long-range connectivity between different brain regions. And also the ultrastructural resolution, that is the synaptic contacts. Now, what are the imaging modalities that can provide you with data in these at these different scales? So in the clinical domain, we typically have magnetic resonance imaging, MRI, which is used both for structural connectivity scans. This is diffusion-weighted MRI tractography, but also for functional connectivity, which can be understood as correlation between neural activity in different brain regions, and this is facilitated with fMRI. At the mesoscale, we have light microscopy, and with these light microscopical techniques, you can visualize individual neuronal cells. This is actually an image measured by our cooperation partners in Bonn, which is showing one cell with a soma, and these thick areas are the dendrites. You can see synaptic spines, these little protrusions, and these very fine branches are axons, which are forwarding signals to other neurons. And if you want to identify the synapses, actually, there are techniques to do this with light microscopy, but to have really clear resolution of the synapses, you're actually tied to electron microscopy, which makes it much more complicated to actually obtain data, particularly large tissue volumes of data that give you some ground truth synaptic connectivity. All right. And this EM-based resolution is often also called ultrastructural resolution, because you can really, these scans are at resolutions of usually like 4 nanometers. So you really can see the structure and the shape of individual synapses with these imaging modalities. So one exciting development in the availability of these data sets is that more and more this gap between the light microscopical world and the electron microscopical world is bridged. Here we see a recently published data set about the human temporal cortex, which is a tissue sample from a human brain, which was excess tissue removed during an operation. And it was reconstructed by Lichtner Lab and was made publicly available. And here we now see that this huge EM data set covers entire dendritic trees. And if we zoom into this data set, we will see that indeed we now have the full scale resolution from, say, the mesoscale, the extent of entire dendritic trees down to individual synapses. So these are mostly excitatory neurons. It's a small subset of the neurons in this data set. It's much more dense, but it would be totally cluttered to visualize it all together. And we've now zoomed in to the scale of individual synapses, and this orange sphere is the location of one synapse, where this blue axon is connecting to this red dendrite. And you see now in the foreground these protrusions, dendritic spines, which are the main postsynaptic connection points in excitatory neurons, and they'll be of relevance later for our models as well. Now, we started with a light microscopical model of the red barrel cortex. This has been the domain in which our cooperation partners in Bonn have been extensively working over many, many years. What is the red barrel cortex? It's an area in the somatosensory cortex of the brain, and it processes sensory information from the whiskers in the snout of the rat. And the distinct feature of this brain area is that these whiskers in the snout of the rat have a somatotopic representation in the cortex. It's a somatotopic map, which is shown here on the left. And this is preserved across animals, providing anatomical landmarks to actually study this brain region in detail and also make comparisons between animals. What we see here in the zoom out is, if you dived into, we're now looking on top of the cortex, but if you dived into the cortex, you would see these 6 cortical layers that actually implement the signal processing circuit, with layer 4, the main input layer, which is then propagated up to layer 1 and 2. And layer 5 represents the main output layer, and complex circuits are implemented within these barrels. Barrels are these columnar structures, one barrel corresponding to one whisker, that implement these signal processing circuits for individual whiskers. The goal of our cooperation partners in this project is to build a detailed in silico model. And one could ask why we want to build an in silico model of this brain region, why we want to build an in silico model of the red barrel cortex. And as indicated, one reason is that by building a standardized averaged model of a brain region, we can account for inter-animal variability that is found in experimental data and get sort of a standardized representation of this brain region. Second, we not just want to understand the wiring diagram, but of course the goal is to understand how this circuit works. And for this, we need to simulate neural activity. And to simulate neural activity, we need realistic constraints about the wiring diagram. And these constraints can only be derived in a computational format if we actually have a computational representation of this brain region. And also, we want to develop hypotheses in silico that are then used to make targeted experimental measurements and vice versa. They can help to better understand experimental measurements if empirically observed structural or functional features can be replicated in the in silico model. One final and important reason to build in silico models of such brain regions is that current imaging techniques, including EM, are still limited in what they can provide. For example, at these non-invasive macroscale imaging techniques, of course, we don't have single cell resolution. And in these EM scans of neural tissue, we are currently limited to very small volumes of neural tissue, which makes it difficult to understand cellular connectivity between different layers or different columns, for example. This is also illustrated in this chart, where we see that these axons of neurons extend well beyond the location of the soma into surrounding tissue. And in these EM scans of neural tissue, we are currently bound to small, thin slices. And of course, inadvertently, we cut off a huge part of these axons and lose connectivity information. This is also seen here in this chart from this human temporal cortex data set, where we see this light blue dendrite, which is within the slice, but we see that the axons cross from one side of the slice to the other side of the slice. And of course, they can't be traced back to their parenting soma, and we have no information what the role of this axon is. And in this context, sort of this light macroscopical approach still has its advantages, because here we can label individual cells. That is how we retrieve the morphology samples. Here we see an illustration of that. This is one neuronal cell. Red is the dendrite, and blue is the axon. So this cell was labeled with a marker in the neural tissue, and then histological sections of this tissue were light microscopically scanned and then reconstructed to get this line structure of the neuronal branches. And here visualization plays an important role. At SUS Institute, we have developed the Amira software, which is used to edit these line structures, so you can do tracings in imaging data. And a very important part in so there's semi-automatic pipelines, of course, to retrieve this stained neuronal structure from the imaging data. But in all cases, proofreading these morphologies is required. And over many years, we have developed features within this software that enable the proofreading of such morphologies. And they're extensively used to arrive at these neuronal structures. Now, with this approach, we can now collect a set of representative samples of cells from different cortical layers and spatially register them in the volume of this barrel cortex model, and then get a sort of artificial in silico representation of the wiring within this model volume. One thing to note is that the packing density in these cortical areas is incredibly dense. So what we see here are only the somata, not the axon and dendrite branches. And the soma distributions have been experimentally determined, also by staining tissue slices across cortical layers. And we have then averaged over 4 such measurements the typical soma density. And within the model volume of the barrel cortex, we find 500,000 neurons. And one important aspect is that the distribution by cell type, here on the right, we have colored these neurons by cell type and cortical layers, is highly heterogeneous. For example, here we see in these green areas these barrels where the composition of cells is actually different than in the surrounding septa in layer 4. If we do an upscaling, we now map these representative morphology samples to every soma, to every cell body within the volume, and we obtain a dense model. And in this dense model, we now have a realistic packing density as it would be found in neural tissue. And here are the components of the model. So we see the somata across the layers, the dendrites and the axons, which is almost too cluttered to really identify it because they're so extended and dense. In total, you would find 25 kilometers of cable within this model volume. And if you pick out this small cube, this 100 micrometer times 100 micrometers times 50 micrometers, you would find 2 meters of axon, 30 centimeters of dendrite, and 500,000 synapses in this volume. Now, this gives us the composition of the branches, the axon and dendrite branches. But of course, we're interested in synaptic connectivity. And also over many years, a connectivity model has been developed, which is based on overlap of axon and dendrite branches, but also on subcellular features. What do I mean by subcellular features? The presynaptic connection points of parts of a synapse are boutons. So these are extended areas along an axon. And there have been experimental measurements conducted to count how many boutons you find along these axons. And the boutons, not the synapses, are still visible in light microscopy. So we have experimental measurements of the bouton length density. So for all of the axons within the volume, we can estimate the number of boutons that you find spatially. We can do the same for excitatory postsynaptic spines. So these are these little these structures which are marked here by these red arrows, and so we also obtain a length density for the spines. And particularly at inhibitory neurons, which is a different cell type. Two broad classes of neurons are excitatory and inhibitory neurons. And inhibitory neurons are usually connected by targeted by axons at the surface area of either the dendrite or the soma. So based on the extent of these line structure of these reconstructed morphologies, we also obtain estimates about the surface area. And based on these three subcellular features, we now have an estimate of how many pre- and postsynaptic connection sites are available in any, we call it overlap volumes, which are small cubes that partition the entire volume. In each of these cubes, we then compute for every neuron pair the estimated product of presynaptic sites, postsynaptic sites normalized by the overall number of postsynaptic sites, and using a stochastic model, assuming a Poisson distribution based on this overlap quantity, DSO, then structural overlap, we obtain an estimate for the number of synapses. We can do this for every cell in our model volume, and as a result, we get a very large connectivity matrix, which gives us predictions, stochastic predictions, about the to-be expected synapse count between any cell pair in the model volume. And we see this here. This is the zoomed out representation of the matrix, where we have this grouping into rows of the Whisker field. And if we zoom into one of these rows, we now have one, this is the C2 column. So this would be this barrel. And within this C2 column, we have now grouped the rows and columns in this matrix by cell type. And for any pairing of cell types, we can now also retrieve the distribution of predicted connection probabilities. And this already gives us some structural insights. For example, we can use this model to assess how connectivity patterns would look like in this model if we have this stochastic, essentially parameter-free, I mean, there are empirical quantities going into this model, measured quantities, but essentially it's a parsimonious parameter-free model. And based on this parameter-free model, we now get a baseline or a null model that gives us a first-order estimate of what connectivity would look like solely based on these morphological structures. So this is the structural insight from the model, that although we only used morphologies in a very limited set of empirically measured quantities, the connectivity patterns that this model produces are already highly non-random, suggesting that morphology actually has a considerate impact on the connectivity structure in such cortical networks. But having access to these connection probabilities is also relevant for another reason, because as said, we want also to simulate neural activity. And what we can do now is to take one given cell that we want to model in detail. Here we say layer 5 pyramidal tract neuron dendrite, place it into this dense model, and based on the connectivity predictions, we get a understanding, or we get anatomically constrained data of which synapses are likely formed by the surrounding cell populations. And this complex model, consisting of point-like presynaptic neurons surrounding this dendrite and the full compartmental model of the dendrite, allows for very detailed neural activity simulations that can then be compared to experimental measurements of neural activity to actually understand or better understand how network function is implemented in these circuits. Finally, I want to point to Barrel Cortex and CDCO, which is a web application that provides access to some of the data in this model and also provides some interfaces to query the model and the morphologies. Now, this was our this sort of concluded work about the red barrel cortex based on light microscopical reconstructions of morphologies. I also ventured, or one aspect in this cooperation project was to build generative network models. In our context, a generative network model is any model that predicts synaptic connectivity using a stochastic approach, using first structural features, as we have seen, the number of these boutons, the number of these spines, and they're also related to the branch length, for example. But to extend this, we can also integrate more complex assumptions about synaptic wiring preferences. For example, it is known that certain cell types in nature preferentially connect to certain compartments or have that connectivity is not entirely random. There is targeted connectivity found at the subcellular scale in such cortical networks. And refer to these free parameters or these specificity parameters as synapse formation strategies. And for now, let's denote them by some free additional parameters alpha. And our approach is now we assume that we have a dense reconstruction, which could either come from a model or even better, EM scan, which gives us the subcellular anatomy in one of these little overlap volumes. And we now sort of shake this box and dissolve the empirically observed connectivity between these cells in this overlap volume. And we can now test different assumptions. And the idea or the advantage of these generative models is that you can plug in different assumptions and compare the output of the generative model to some ground truth. This ground truth could be empirical measurements that are sparse, for example, using patch clamp recordings, where you only have access to certain very limited set of cell pairs that have been measured. But also, even better, if you have dense reconstructions, you have a ground truth connectome, and you can compare these model predictions about synapse formation to the ground truth connectome. And this is illustrated by these little matrices here on the bottom right. So we remove the ground truth connectivity, we retrieve from this structural reconstruction anatomical features, and we now let the model operate, and the model could output different connectivity predictions based on the parameters that we input. Then we do a comparison to the actual connectivity, and we can learn which parameters work and which don't, and thereby understand which synapse formation strategies are necessary, best suited to explain connectivity at this subcellular scale. And we now wanted to apply these methods to datasets that are currently available, and as indicated, more and more large-scale dense reconstructions of neural tissue are becoming available. Here we see an illustration or a rendering of a subset of the neurons in this mouse visual cortex reconstruction. So we see these cell bodies and then a very complex network of branches from these cell bodies. And if we would zoom in, as we did in this initial video illustration, zooming into the human temporal cortex, we would see the ground truth synaptic connectivity. And this is what we need to apply our models and learn about synapse formation strategies. A bit more formally, so what is the input to this generative model? We have a ground truth connectome denoted C0. We define a stochastic generative model, which gives us a connectome given certain parameterized synapse formation strategies alpha. We sample parameters of this for the synapse formation strategies. And then we use a Bayesian inference approach to infer the posterior distribution of parameters, that is, the likelihood of alpha parameters to regenerate the empirically observed connectome. And there'll be an illustration later that makes that clearer, I think. But let's first take a look at the synapse formation strategies that we are currently modeling. So these boxes here on the top show 3 categories of synapse formation strategies. So the first category, the alpha P category, simulates wiring preferences between different populations of neurons, for example, excitatory neurons and inhibitory neurons, but it could also be layer-specific wiring preferences, for example, layer 4 neurons to layer 5 neurons. The second group of synapse formation strategies simulates wiring preferences or molecular recognition between individual cells, so that one specific cell pair would recognize each other and have some preferential wiring pattern. And the third category, the subcellular alpha S category, simulates wiring preferences that refer to which postsynaptic compartments synapses are established. And here usually we distinguish synaptic spines, apical dendrite, basal dendrite, the soma area, and also synapses can be established at the axon initial segment at postsynaptic neurons. And how this model currently operates or how it's implemented is that we iterate over all these little cubes in the volume. Within the cube, we have this neuron morphology from our dense reconstruction. We decompose it, removing the synapses, but keeping the structural features, such as which cell types innervate this overlap volume, for example. And then the parameters in the model are we call it specificity parameters. These are the alpha parameters. Currently, they are at a scale of -1 to +1. -1 indicating repulsion or unpreferred contacts between these two sites in the overlap volume, and +1 a preferred or attractive contact between these volumes. And currently, our approach is to add these different specificity parameters up and then use a softmax function to get a normalized, row-wise normalized specificity matrix, which can be interpreted as probabilities that any contact in this overlap volume is connected to each other. And based on this connection probabilities at the local level, we can then generate with the stochastic process this forward model, the probability to get a connectome given certain wiring specificity parameters. And of course, the idea is to now test different combinations of specificity mechanisms. So here we see the alpha 0. This would be our null model. So this would be the assumption that there is no wiring preference at all in nature. And these other combinations then add more and more specificity mechanisms. And this would represent, in our current version, this would represent the most complex specificity model. One of the technical aspect that we've been working on actually it's a key ingredient of our approach, has been developed together with the lab of Jakob Macke and Jan Bölz, where we're using Bayesian inference to now actually obtain these alpha parameters. And we sample, to do this, we sample from a prior distribution of alpha parameters. We use our forward model, explained in the previous slide, to generate a batch of rule-generated connectomes. And we then use a neural density estimator, which is essentially a neural network that learns, in our case, a Gaussian mixture density distribution, to predict the posterior distribution of our parameters. That is the probability of alpha parameters to regenerate the empirically observed connectome. And starting from a uniform distribution in the Briar, we don't know which parameters actually work, which don't. With a very limited set of simulation runs, we can arrive at a posterior distribution which tells us which parameters are most likely to reproduce the empirically observed connectome. And the advantage of this method is that we're not trying to optimize one single parameter to optimally fit the data. What this output gives us is the entire distribution over parameters that are data compliant. Because we have this probabilistic approach where the insight comes from the posterior distribution over parameters, which gives us the probability that these parameters can reproduce the ground truth data. Now, applying this to the mouse visual cortex, here are some preliminary results. I think the details of these bars are not too important. E stands for excitatory neurons, I stands for inhibitory neurons, and these subscript letters DSA stand for dendrite, soma, and axonal initial segment. So here we see all possible combinations in the model between which cell populations synapses are formed. And what is shown in black is the empirically observed connectivity. Oh, by the way, these U parameters, U cell groups are unlabeled neurons. And in fact, one limitation of these data sets is that most of the neurons are in fact, or most of the synapses that you find in these reconstructed volumes are in fact not traced back to a soma, so they're not tied to a specific cell type. And the gray bars are our null model. This would assume that we don't have any wiring preference. And what we see is that this null model has some qualitative correspondence to the empirically observed data, but it also fails to capture certain aspects of connectivity. For example, we see that the null model systematically underestimates the number of synaptic contacts from excitatory neurons to the dendrite of inhibitory neurons. However, the null model overestimates the number of synapses from excitatory neurons to the soma of other excitatory neurons. And if you repeat this for all combinations, you can now infer wiring specificity parameters that indicate certain wiring preferences. And we've also done this for this statistic, which shows the number of synapses, that is the number of synaptic contacts per neuron pair. So the 2 here indicates that any pair in the is connected by 2 synapses rather than 1, and so forth. And well, currently the takeaway from this slide is that it's a sort of a proof of principle that we can fit the data with this approach. I'm still very cautious about biological interpretations at this point. What we also can do with this approach is to run this inference not at the group level, but run it at the level of individual cells. And this has been done here. So here we see one excitatory cell and one inhibitory cell with its connections. Here, these triangles represent other excitatory cells and these circles inhibitory cells. And we can now let the model fit the optimal wiring specificity parameters for these individual cells, and then we get a distribution over the inferred wiring parameters for individual cells, which can then be used. So in this model, we did not provide the information of the presynaptic cell type to the model, and we can now infer this information back from the model. So for example, we see that there's a very clear distinction between excitatory axons and inhibitory axons, although the model, when we trained it, didn't know about the identity of the presynaptic cell type. And based on these distributions and differences in the inferred wiring parameters for individual cells, we can arrive at a dissimilarity matrix and use this to generate a clustering of cells that separates more or less the excitatory and inhibitory cells in this model. This concludes this part about generative models. I should stress it's still ongoing work, and I just wanted to share my current results. Another aspect that we're dealing with in our lab is interactive visualization. The idea of interactive visualization is that you support scientists, or in this case, our domain experts, the neuroscientists, by providing visual interfaces that facilitate their workflows. And as we've seen initially, this started quite a while ago with our Amira software, which has been used for proof editing mainly, these neural morphologies and constructing the structural model. And based on the structural model, we can now do biophysical simulations. And both to understand the structural model, we apply visualization to better understand the network structure in the structural model. And we also apply visualization, interactive visualization, to understand the parameter space of these biophysical simulations, because it turns out that you can imagine you can make these biophysical models arbitrarily complex with many, many parameters that describe, for example, the ion channel expressions along this dendrite. And then the question is, how do you find meaningful parameters and how do you understand your parameter space? And this is where interactive visualization comes into play. I'd like to illustrate this with two examples. The first was a design study called the stratification matrix. So here we see a tabular structure, which is simply for illustration, where we have a patient population of N100 grouped by two axes: gender, age, treatment type, and treatment outcome. And this is sort of, I think, the origin of this term, that you can stratify populations based on properties. And technically, it's defined as organization into hierarchically ordered attribute layers. As said in this example, a stratification of a patient population into hierarchical subpopulations. And we now wanted to build some flexible interface to visualize such stratification matrices, where you can group a population based on their attributes, which can be freely assigned also hierarchically to rows and columns of a matrix. And in our case, these entities that we want to stratify based on their properties are biological entities, namely neurons and synaptic connections. So one example is shown here, where we use this tool to stratify the neurons in our barrel cortex model, both by morphological cell type, which we assigned to the rows of the stratification matrix, and by cortical depth, which is here binned into increments of 100 micrometers. And then we see this layered structure of the distribution of cells in this model. And another scenario is that we use this tool to visualize the distribution of synaptic contacts. Here we have the synaptic contacts in the model stratified by layers. Rows in this case are the layers in which the presynaptic neurons reside, and the columns are the layers in which the postsynaptic neurons reside. And the matrix cells here show the number of synapses between these neurons. And here we added a split glyph, which allows also comparing different data sets. And the data sets we compare in this case is the ground truth versus a model predicted synaptic connectivity count. And if you zoom into this matrix, you can get a more fine-grained resolution. Then these column cells will split up, and you get a more a grouping also based by other properties that are that have been assigned to the rows or columns. Another design study that we did deals with coordinated multiple views. That is essentially an established concept, coordinated multiple views, where you have different views that allow the user to make selections, and the selections are then propagated to other views, a technique that is also called cross-filtering or brushing and linking. In this design study, we actually looked at how we can define such views using heterogeneous JavaScript libraries and a grammar-based specification language. But what we see here is actually one use case where the tool has been used to study the distribution of synapses and the precise location of these neurons. And here we now see this example from the biophysical parameter scenario, where we use this tool to identify parameters that in this case facilitate energy efficient dendritic computation and better intuitively understand the parameter space. And yeah, we also experimented with views where you can visualize the simulation outputs and then inspect and compare different parameter samples. Now, currently ongoing work is, which I'm doing with Jakob Treudel and Michael Riga at the Visual Computing Group at Harvard, is to try to embed such visual analysis tools into Jupyter notebooks, because one key insight also from previous projects was that we need to really enable the adaptation of these tools into scientific workflows. We probably should shift to Jupyter because it's mainly used by domain experts and in the data mining community and neuroscience community. And the ecosystem in this area is developing rapidly. So it's now possible to embed also 3D viewers into Jupyter notebooks. And we are currently working on a viewer that facilitates the display of neuronal structures, and the comparison of neural structures. Actually, the background is that we want to compare data sets in the fly brain. We have certain connectomes coming out from the fly brain, male and female, and we want to make comparisons between these connectomes. That's why it's called CompCon. But this is still ongoing work. But takeaway is that we want to shift this from web-based tools to Jupyter notebooks. This already concludes my talk. So my conclusion is that, well, what I wanted to convey is that anatomically detailed brain network models are essential in computational neuroscience because they allow to study networks beyond the scope of what is currently possible with experimental imaging techniques. They are required to run realistic neural activity simulations. They allow neuroscientists to develop hypotheses about network structure and function, which then inform experiments. The second point is that interactive visualization facilitates the scientific workflow, as we've seen for the model creation, this proof editing, but also for explanatory data analysis, and in many cases also for the communication of results, particularly if they involve 3D data sets. And as indicated, future VIS tools or ongoing work is to port these VIS tools to the Jupyter environment, because that will simplify their adaptation. Here are the references. And finally, I'd like to thank you for your attention. Thank DFG and SPP 2041 for the funding. And I'm very happy to go into questions.
Philipp, many thanks for your talk, full of interesting implementation details. And now we have time for questions, answers, and for discussion. So, I open this part. Before the first question will arrive, I would like to ask you what do you think about validation of these approaches? How it can be, how all these approaches can be validated in a convincing way?
Well, for the barrel cortex model, we actually ran some validations using experimental measurements, for example, patch clamp recordings, but also EM recordings at the EM measurements at the subcellular level, to get a understanding how well the model approximates empirical connectivity measurements. So this gives us quite some confidence that the model predicts realistic connectivity patterns.
Mm-hmm.
But I totally agree that validation of models is always a critical aspect, and you never have a ground truth. Even if you have a dense connectome from a EM reconstructions, that is your n is one, that represents one specimen. And the question is, what is the ground truth in some average model? Then maybe it also referred to the visualization tools that we typically have structured ways to gather feedback. But also in this scenario, I agree that it's always a debatable point how you can demonstrate the utility and the benefit. Yeah.
Yes, and I understand that this is what is the most probably the most important and the most significant contribution of your work is elaboration of a kind of approach, a kind of methodology, because it can this validation can change from person to person. Yes, so it is we do not have something like universal Brian, as I understand.
Mm. Yeah.
Okay, we have Wiesław Nowinski with us, and Wiesław is going to take part in the discussion. Wiesław, you can ask the question.
Okay, thank you, Marian. Can you hear me?
Yes.
Yes.
Okay, Philip, it's exciting work. Congrats. Now I have a couple of questions. What is the highest challenge and fun doing this project?
Sorry, I didn't understand you acoustically, the last part.
What is the highest challenge and fun doing this project?
Oh, the highest challenge, I think, is rooted in the data sets that, particularly in these CM data sets, we are still dealing with a lot of reconstruction artifacts. So we are quite grateful that they're now publicly available, but one continuous challenge is to fully trace axon morphologies, and these axon morphologies are quite critical for cellular level connectivity. And the second part was the most fun part of the project, correct?
Yes.
Okay, yeah, well, that's
What do you like the most?
yeah, that's definitely the visualization part, because there's some artistic element to it, and usually you can also prototype these tools with your users, and that's the fun part for sure. Yeah.
You mentioned that there is lots of variability in experimental data. Can you quantify the amount of variability? Is it 5, 10, 50%?
I would be hard pressed to quantify it. But typically, I mean, it starts with the fact that usually these connectivity measurements are based on single cell pair recordings. And that is essentially where individual cells are probed. And then you do this for multiple times in patch clamp recordings, and then count how many cells are connected. And the first part is to really map the cells from experiments to the cells in the model, because in some experiment you don't have the exact layer, the exact depth, and there's no standardized, fully standardized way to really come up with exact locations. So the first variability is already in the experiment. How do you map the experiment to the model? And then in the connectivity measurements itself, there is significant variability between certain studies, but I would be hard pressed to quantify it.
I see. As far as I understand, the current work is limited to sensory cortex, yes? Because you mentioned about whiskers, the visual cortex.
Mm-hmm.
I think it's only limited to the cerebellum. Do you also plan to do any work regarding the complete cerebellum and cerebellum?
Then we have to distinguish whether we are talking about the barrel cortex model that is indeed sort of all focused around the barrel cortex. All the data has been curated for this brain region. But for our generative modeling approach, we're trying to infer synaptic wiring specificity parameters. This should be not limited to any brain region at all. So we are currently also working with someone who has data from, is also the mouse visual cortex, but a different layer. And that would be very interesting to actually apply this to other brain regions as well. So all we need essentially as input to this model is a dense reconstruction, which gives us these anatomical constraints, that is the overlap patterns and the synaptic connectivity. Once that is available, say from some EM reconstruction of a brain area, we can apply this model. We can also apply this model if there's not a EM reconstruction available, but a light microscopical reconstruction, which gives us the distribution of cells and morphologies. But then we need some additional data to constrain the model. These are these empirical connectivity measurements, for example, from PET recordings. So short answer, it should be the generative model should be applicable to other data sets as well.
Based on the current work, is it possible to estimate how many microcircuits are in the rat or mouse brain?
Let me briefly jump back to the visualization of the model. So by microcircuits, is it do you mean the number of certain connectivity patterns or motifs, or I mean it's I mean the total number of synapses that you would find in this kind of area is in the billions. So even in this small volume, you have 500,000 synapses. So it's a highly complex network, which is also illustrated by this connectivity matrix. So you can have arbitrarily complex circuits in there. But of course, there are certain patterns which have also been extensively studied in the past. For example, this signal flow from into layer 4 into the upper layers, and then layer 5 is the main output layer. So there are certain patterns, but it's hard to give like a number of the number of microcircuits.
Yeah, and this is what I thought that. But I thought that you have some maybe initial approximation. What about talking about numbers? What about numbers of synapses per neuron? I think on one of the slides you show that it might be variable from 10 to 1 million. Any numbers here?
So here it's broken down to the number of synapses that we find in the volume. The number of synapses per neuron strongly depend on the size of the neuron. First, you have the size of the dendrite. This is closely related to the number of incoming neurons. And then you have the length of the axon, which is closely related to the number of outgoing synapses. For example, in these layer 4 cells, you would expect less incoming neurons than in, say, some much larger cell. So it's highly cell type dependent. To give a rough estimate, it's in the tens of thousands for these layer 5 pyramidal tract cells, the number of synapses per neuron.
Another issue you haven't mentioned, but it might be of interest of Sano. Have you or maybe your consortium consider microvasculature?
It's just to confirm, this is about the blood vessels in these areas, correct?
Yes.
I myself haven't studied it. I think our cooperation partners also haven't delved into this. But I know that in some of these recent EM data sets, for example, of the human temporal cortex, but also recently there was a release of a fly brain data set. these vascular structures have been reconstructed and can be visualized and can be studied. And I think there are people that are actually, that there should be data sets available that that provide you with information about these vascular structures. I'm not sure about, it depends, of course, on the brain region. I'm not aware of any such data set for the mouse, for the red barrel cortex, or the for the red barrel cortex. So it depends on which area would be interested in.
Yeah, because it looks that this week we'll be getting data for microvasculature for monkey. So meaning at the
Okay.
level of microns,
Mhm.
synchrotron CT. And I
Mhm.
believe that many challenges which you faced, we might be facing in the future. For example, can you comment more about proofreading? How you are doing proofreading? I believe that we have the same complicated network after reconstruction, segmentation, and visualization. So proofreading, I think, will be critical. So your experience here will be,
Yeah.
I believe, very, very valuable.
Yeah, currently, so Amira, in this project, Amira is used for proofreading. I should mention there are multiple softwares available, of course, but I can certainly recommend Amira for this as well, which has a filament editor where you can sort of pick individual segments of the neuron and remove connections, add branch points, remove intermediate branch points. And particularly in this project is the challenge is that we have imaging data from histological sections, and we have a semi-automatic pipeline which gives us most of the neuroids, but there might be gaps between different segments, and then these gaps are filled, can be filled, or can be corrected manually. Also, what Amira allows is adding, which is not shown in this image here or this video here, is adding an image layer to the on top, which is like you see it here on the left, an image superimposed on this line structure, and then you can pick certain areas on the image and shift your image to areas of interest where there might be gaps in the or reconstruction artifacts to get an estimate. Well, to manually compare the image to what the line structure looks like, and then make corrections if necessary. But it's certainly the whole proof editing is a huge field, and there's many recent works as well in, particularly in this EM domain, very comprehensive tools that facilitate proof editing. So it's definitely worth to do some in-depth search for the available tools. Yeah, as indicated, we use Amira. Yeah.
Okay. Yeah, and maybe at the end, taking at the pace of progress, how do you think when we'll be able to reconstruct the complete connectome of red? And even more for the human, what do you think?
Well, the question is, what do we mean by reconstruct? I mean, I think that the image acquisition volume is shooting up, and also the automatic proof editing algorithms are getting better and better. But what we see in currently available data sets is that there are still very many proof reconstruction artifacts, particularly in the tracing of these very thin axon structures. And I think this is still an open problem. And I think the bottleneck, should mention, I'm not too deep into the proof editing field, but my estimate is that the bottleneck currently is actually the man hours for the manual proof editing in this field, because it's still required, as per now with current techniques, to have a manual proof editing step. And I have the impression there's more data than can be proof edited. So it's that is still a bottleneck, but who knows? Maybe the algorithms will get will outperform humans at some point, even in these manual reconstruction steps. And I mean, I think my latest understanding was that there is an EM data set covering one cortical column in the red barrel cortex. But I think a dense reconstruction of the entire mouse brain is still far away. I think that the current milestone, I should say, is also been currently released is a complete reconstruction of the fruit fly plane, but that is in the range of 130,000 neurons. For comparison, in this small area in the rat brain, we already have 500,000 neurons. So this gives you an estimate for an impression of the scales that are still differing. I should also mention that there are, for example, on Blue Brain, there are approaches to indeed reconstruct the mouse brain, but then not at a saturated level, but also based on light microscopically reconstructed morphologies. So it always is a question, what kind of imaging modality do you use and what kind of resolution do you need for your analysis? And there is not yet a silver bullet with current imaging techniques.
Okay, thank you. Enjoy, have fun, and lots of successes.
Many thanks for both of you for interesting discussion. Now we have in queue Professor Witold Zwinel from Institute of Computer Science. Witold, you can talk, I think.
I think Witold left the seminar.
Witold left the seminar, but there was a question. Does the same or very similar generative part of the model can be used for modeling other complicated biological graphs like vascular network in various tissues or whole system consisting of complicated tissues mixture, regular tissue, cancer tissues, something like this. To which extent your approach is universal?
That sounds exciting, but I think I mean it would be exciting to try this. And I mean, generative models are a very general concept, so they're not restricted to neural networks, biological neural networks. But I haven't explored this direction.
Yes, so probably it is what you have done. It is just well suited for reconstruction what we have in our brains. Yes, so I think that it.
Yeah, in fact,
yes,
I think it's very targeted currently. The whole formulation of how we model connectivity to this domain. So yeah.
Yes. Well, at that moment, I do not see more questions and people willing to discuss. But I would like to thank you very much for interesting talk. And I think that, also many thanks to Professor Zachow for advising to invite you, because I think that it could be not just one meeting, not just one talk at the seminar, but I think that in Sano we have people like Jean, who was talking a week ago, and I think that it will be possible to, let's say, establish a kind of collaboration, just exchange ideas. So I hope that it will be not just this one talk. Thank you very much.
I would be excited to catch up at some time, and thank you again for having me.
Thank you very much for being with us. Many thanks. Well, and I would like to invite all of you to next seminar, which will be as usual on Monday at 2 o'clock, and it will be next time will be more about let's say computer science, but anyhow, computer science related to computational medicine. So thank you very much, and goodbye, and see you in a week from now.